Now, continuing on with the next module, 9.1 and 9.2, we continue with cancer. And for cancer, just take note that these are abnormal cells that divide without control and are invaded or invades other tissues. So these are the important characteristics. And take note that there are three important external factors that will be influencing cell division. Number one would be growth factors. Number two would be density dependent inhibition. And the third one is anchorage dependent. So when I say growth factors, just take note that the normal cells with growth factors they will divide, but without growth factors normal cells will stop division. But the problem with cancer is that with or without the growth factors, the cancer doesn't care. It will continue on dividing and dividing and dividing, so that the tumors are produced. On the other hand, the second external factor is a density-dependent inhibition, and in this case, it simply means that when there is not enough space, normal cells will stop dividing. But for cancer cells, even if all of the spaces has been used up, because it is not dependent on the density and it doesn't care, so what will happen here is that the cancer cells will continue on dividing and dividing and dividing, even if there are no more space for the cells. So that's the characteristic of the cancer cell. That is why it produces tumors. On the other hand, anchorage dependent simply mean that these normal cells would divide if it is attached to a surface. When it is not attached to a surface, the normal cells will not start dividing. However, for cancer cells, again, it doesn't care whether there is a surface to attach to or not. It will divide and divide and divide, causing the production of tumors. So those are the three important external factors: the growth factors, the density-dependent inhibition, and the anchorage dependence. Now, do remember that cells or cancer produce our characteristics are irreversible, and then it is also gradual multi-step, which means you don't produce cancer with just one mutation. You produce cancer with the accumulation of mutation, as you can see over here in the drawing. So there are two important genes that you have to take note of that produces cancer. The first one is proto-oncogene. Proto-oncogenes are actually very important genes because they are important in normal cell division. However, because of certain mutations that happen to proto-oncogene, they become over-aggressive, overproduce, causing accelerated and increased cell division, leading towards cancer production. So there are three ways by which these cancer genes, the proto-oncogenes, become oncogenes, and the first one is what we call as translocation or transportation. So the gene is moved to a new locus. Perhaps it is attached to a new promoter that is over-aggressive, thereby producing a lot of the proteins, growth stimulating protein, causing the cells to accelerate in division, producing the cancer. So that will be the first one, translocation to a overly aggressive promoter. On the other hand, there's the gene amplification, and this means that because of certain problems with crossing over and recombination, you not only have one, but you increase the number of the genes. So there may be a second gene or a third gene, thereby increasing also the amount of proteins, growth stimulating protein, which also accelerates very, very much the production of the cell division, leading towards cancer. So that's the second production of oncogenes. The third one is wherein the proto-oncogene is not amplified, it is not even translocated, but rather mutation happens on the gene itself, causing it to be overactive, over aggressive, which means it produces more of the proteins needed for cell division, thereby causing the production of cancer as well. So those are the three important ways by which proto-oncogenes are converted to oncogenes. Okay, the second one would be the tumor suppressor gene, and as you can see already in the name itself, that its very important function is for it to suppress production of tumor. So our body naturally suppresses tumors by using the immune system to cause them to die, or perhaps the cell itself would be recognizing causing apoptosis. So that would be the natural function of the tumor suppressor gene. But the malfunction of the tumor suppressor gene can cause the production of cancer because the absence of tumor suppressor gene would not be able to suppress the tumor anymore. Therefore, more tumors are being produced. So that would be the problem. So you're familiar with what tumor look like, and we are also familiar with two types of tumor. So you have the benign tumor, which doesn't invade or go to other parts of the body, and you have the malignant tumor, which would be transferring, moving from one place, one organ to another in the body. And the malignant tumor is the more dangerous type compared with the benign. So the benign doesn't move, while the malignant tumor would be spreading through other parts of the body, which we call as the process called metastasis. And this is one of the most dangerous, actual dangerous kind of cancer. So there are several cancer treatment we already discussed in class. So hormone therapy makes use of hormones to destroy the cells. Surgery, you remove the cells, the cancer cells from the body. Bone marrow transplantation, so you make use of a very big needle to remove the bone marrow, or to suck out the bone marrow, which will then be placed into a donor, or into a patient wherein, for example, leukemia is the type of cancer. So the new bone marrow, the functional bone marrow, will be able to supplement, to replace the malfunctional bone marrow, hopefully in the patient. For chemotherapy, you make use of chemicals to destroy the cells, but as we have mentioned in class, this will be just like a shotgun that will hit both the good cells and also the cancer cells. Targeted therapy, on the other hand, is better because it makes use of receptors on the surface of the cancer cells, and the molecular medicine recognize these receptors, thereby killing only the cancer cells. So this is one of the more recent advances and techniques that are seeing a number of successes in its application. Radiation therapy makes use of radiation to kill the cancer cells, but just like the chemotherapy, it also destroys both the cancer cell and also the normal cells, but this is more accurate compared with chemotherapy. And finally, immunotherapy is making use of the patient's own immune system, enhancing the immune system to destroy the cancer cells. So these are the different cancer treatment options.